Thank you very much for the introduction and for arranging my lecture here. I should start by saying I'm a very old-fashioned chemist, and so most of the lectures I give, including one course that I teach right now in electrochemistry, I give on a blackboard and I write with chalk. And I'm not much of a PowerPoint chemist, as you will see from the PowerPoints that I show. So I'm going to have to try to explain what we're doing, and in a way that, without putting the things on the blackboard, reactions or pictures, you will understand at least the basic ideas of our work. Is the sound okay? Can people hear me out there? Yeah. Okay, I'll try to speak louder. So also, I decided to tell you about work we are currently doing that is not yet published, or only parts of it have been published. And it's very exciting to me, and I hope you will be interested too. And so, what we are looking for, put the next one. No, you skipped one there. What we are looking for is an answer to a question I asked when I was a student at City College, the first year I took chemistry, which was, when they describe an atom, let's say they describe the atom of platinum. It has a nucleus, it has many electrons in different levels. And do I imagine that as having similar properties to a piece of platinum that's very large and behaves as a catalyst? Now, as you know, I think platinum is a very highly used material in electrochemistry, and it has some very special properties in terms of catalysis, especially for the sake of this lecture, for reducing protons to hydrogen. It's the best catalyst for that. It was first used by Nernst 200 years ago and shows a very, very fast rate compared to other metals. Other metals, especially mercury, bismuth, lead, don't reduce protons to hydrogen very well at all. It takes a much, much higher potential to get them to reduce at those metals. And so platinum has very special catalytic properties. And I wondered, if I simply had an atom of platinum, would it behave like a bigger piece of platinum and still be a catalyst? And so the idea of the lecture then, and what we've been doing for the last few years, is to try to get single atoms of platinum isolated from anything else, only one atom of platinum, and study its properties. Study it electrochemically so that we can see whether it does behave as an electrode. And it doesn't seem to make sense at all that would be the case. When I asked my professor as a student the question I raised here, how does a single atom of platinum relate to a big piece of both platinum? He said it's totally different, that when you make a big piece, you have a metal, you have lots of nuclei, you have lots of electrons that you can run through it, it conducts and so on. It's nothing like that. But there was no real evidence of that, and so we decided that's what we were going to try to do. Now let me try to give you a little picture of electrochemistry, so when I show some of the slides, you'll understand a little bit of what we are doing. So the first slide shows an electrochemical cell. And I don't see it.
That's okay. I'll do it.
Okay. Oh, no, I do see it. So an electrochemical cell is really just a couple of pieces of metal or carbon connected with a battery up on top of the cell, and where you force current to go through that cell and cause a reaction to occur. And for all of our cells, the electrode we use is called a UME, or an ultra micro electrode. And so what electrochemists have found in the last years is that you can make very, very tiny electrodes, that these electrodes have radii of about 10 micrometers, and it can go down even to the nanometer scale. So the ones we will talk about here are only a few hundred nanometers in size. But they show some very interesting characteristics, that the current immediately goes to a steady state current. There's no transience like you get in a bigger current, bigger electrode. And so you can do electrochemistry at these really very small electrodes. Also, they have a limited number of active sites on them. And so if you have something that needs a catalytic site, there are only a few of those sites on very, very small electrodes. And so what you do in the cell is the UME is on the right, and you pass a current, and you measure the potential of the electrode and the current. And if I pass a current that is out where the total current is controlled by mass transfer of materials in solution, shown in the reaction there. It's shown that the components are going up to the electrode. There they hit the electrode, pick up an electron, and then diffuse away. And the rate at which they get to the electrode depends upon the equation I showed down at the lower part. So you keep moving back.
It just keeps.
Sure, it's this one, yeah. And it's
Sorry, guys.
it's proportional to the concentration of material in the solution, the diffusion coefficient. I think we so it's the equation down at the bottom. That's the only equation I'm going to use in this talk. But what it says is the current is limited by the rate at which material gets to the electrode, accepts an electron, and then goes away from the electrode. And what we're going to try to do is make single atoms on that electrode, pairs of atoms, 3 atoms, 4 atoms, growing them bigger and bigger and studying the very different properties that they have. And so what one can find out from this, the next one, is one gets what in the upper, on your side left corner, is what's called a voltammogram. And that's a plot of the current as a function of potential. And the only current we really care about is that flat current up at the top, the limiting current. And that's the current that's being totally controlled by the rate at which a material gets to the electrode. And as you'll see, the idea of what we do is to bring the concentration to such a low level that I only deposit 1 atom, say, every 10 seconds. And to do that, I bring the concentration in the solution down to femtomolar, so down to about 10 to the minus 15 molar platinum. And then the platinum diffuses to the electrode very slowly. You get currents in the picoamp, 10 to the minus 12 amp region, which can be measured, and you deposit only a few atoms onto the surface. So these experiments were first preceded, before we started doing those experiments, we showed experiments where you could look at very, very small particles, nano-sized particles, and we're able to identify those as I make a solution of these nanoparticles, let's say of platinum, and I do an electrolysis at an electrode that is a very bad electrode for catalysis. So the example I'll give here is, say, the electrode at the bottom. The red atom is supposed to be a nanoparticle, a particle of, say, 4 nanometers big of platinum. And the electrode at the bottom is an electrode that doesn't like to reduce protons. It's what we said is an electrode that's very bad in reducing protons, but platinum is very good. So on the right, you see when you hold the potential where protons are reduced at the electrode, I get a very low current. You see on the right side a very, very low current. But when the platinum electrode goes down and hits that, now it has this one platinum nanoparticle sitting on the electrode, and that electrode, that particle, turns on the hydrogen electrode, and so the current jumps up, as you can see, and that's showing the reduction of proton at a much higher concentration than femtomolar now on that one atom. And so if you have a number of these nanoparticles in the solution, you'll see every time a nanoparticle hits that electrode that I get a potential step which shows that particle landing on the electrode and giving a current, and then next particle lands, and so on. So I can study the collisions of nanoparticles and use this idea of electrocatalytic amplification, that I have an electrode which won't do a reaction, but as soon as a particle, say platinum, hits it, a reaction turns on. And it's that step which has all the information about the rate at which platinum is being used as an electrode. And what we've now changed, I think I have one more. What we've now changed is, instead of having the platinum be particles, already bulk materials, once you make a material a few nanometers big, you have thousands of atoms in it, and it starts to behave like a big piece of platinum. And we'll show here how it behaves that way. But if instead of having the particles land on the electrode as a big particle, we now go and deposit, by the reaction you see on the left side up there, we deposit a platinum atom, then we can do the same trick. We can deposit a single atom by having a very low concentration of H2PtCl6, for example, and that material at femtomolar will only deposit one atom every 10 seconds, let's say. And if I do the current, we'll get the effective rate. We'll modify that statement a bit. And so I make the solution in the left-hand side that has material that will deposit platinum, H2PtCl6. And then I will see, on the average, one atom deposited every 10 seconds. So if I turn on the potential for 10 seconds, I should have, on the average, one atom. If I turn it on for 20 seconds, I should deposit two. if I have turn it on for 30 seconds, and so on. And so I can make single atoms, 2 atoms, 3 atoms, and so on. And so this is the whole idea. Then the electrode is put into a solution with protons, and I can see this large current now that comes up from the proton reduction. And I can see how 1 atom reduces protons if it does, 2 atoms, 3 atoms, and so on. And so that's what we're trying to study. The idea is I'm going to make an atom-by-atom synthesis of a metal, or as I'll show you later, a molecular catalyst. And I will see how it changes as I put more and more platinum atoms onto this. And I'll show you these voltammograms that show this. Now the next slide shows a single atom. So if I put 1 atom down on that electrode and then do this either on a bismuth electrode on the left, which is again a very bad electrode. If you look at this slide on the left, bismuth by itself is the very low current. So bismuth by itself is sitting out there, but it won't reduce protons. Similarly, on the other side, it's a lead electrode. Lead is also not a good electrode for proton reduction. And so the current is very, very low. But as soon as I deposit 1 platinum atom, then I get a current, and I get proton reduction, and I get a much higher limiting current. So the size of that electrode current at the limiting current is a measure of the size of the platinum atom. I can measure the platinum atom this is looking for by using the equation I showed earlier. And where the potential where this wave occurs, so if I look at the potential where this current is jumping up, about halfway up, that tells me how good an electrode that is, how good a catalyst it is. If it was a large piece, that current would be much, much lower potentials, and the same with lead. So that's the basic idea now, that I put down 1 atom at a time, and I study the rate of reactions and the size, so I can get the size and the rate, and then try that with 2 platinum atoms, 3 platinum atoms, and so on. And so the next one shows a series of these now curves with different numbers of platinum atoms. The lowest one is 1, the next one is 2, and so on. And what you can see is the current is, of course, going up because I'm increasing the area from 1 atom to 2 atoms to 3 atoms. And I can show that that's not 2 atoms on different places on the electrode. And that's what I meant by the limited number of active sites. The number of active sites is small, so when 1 atom deposits and the second is going to deposit, the highest probability is it deposits right onto the first atom and the second. And that's why you see the potential getting more and more favorable as you add more and more platinum atoms. The curves are moving over and the rates are going up. And so you can see if I make combinations of platinum atoms, as I go from platinum 1 to 2 platinums to 3 to 4 to 9, it gets faster and faster and faster. And so this answers a question that I said has bothered me for 60 years, that even a single platinum atom, which is nothing like a bulk piece of platinum, will act as a catalyst and will reduce protons or do other things that we study. And then we can do it even more and grow bigger particles. And you can see then how it improves. That the x-axis is the number of platinum atoms, and the y-axis is the rate at which you're doing that, the rate at which you're depositing. And you can see the platinum atom is slowest, but still doing something. And as I add atoms, I go up and up and up and up, And the dashed line up at the top is the rate at a bulk piece of platinum. In other words, once I get to a piece that's large enough that it now is a piece of metal, then I get that steady state up at the top. Now, a question that should be in your mind is, when you say, when I say, 10 seconds per atom, if I'm using large amounts of material in a solution, and then I call it an ensemble of materials, then I have billions and billions and billions of molecules in that solution that behave completely differently than this. I have, the number, if I have 2 grams of hydrogen, the number of atoms I have is more than all of the stars in the universe. So a mole of atoms is really 10 to the 23rd, higher than 10 to the 23rd. That's more than the stars in the universe. And so there are lots. So it's a very different situation. And so when I start to do one atom, now it becomes not this same kind of not the same kind of deposition that I would have with an ensemble of atoms, because these are called stochastic processes. In other words, when I have an atom in a small volume of solution where it's going to take 10 seconds to diffuse across that solution, there may be no atoms in that by chance. There's a probability there are no atoms in that volume. There's a probability that there's 1. There's a probability that there's 2 or even 3. And that's called a Poisson distribution. So when you do these kind of experiments, there is no real single number that can tell you definitely it's one atom. It could be none. So sometimes when you do 10 seconds, nothing happens. Sometimes you get 1, sometimes you get 2. And that's calculatable by just probability theory. And that shows it on these. So if, in fact, I do the same experiment with, say, 10 seconds, then you get a distribution of sizes. Back. Put it back. Just put the slide.
I did. Give me this one.
That's the Poisson. Poisson. Go for it. Yeah, this one. You get a distribution shown in one color, and the other color is the actual experimental results for a finite number of trials. And what's interesting is it follows that distribution very nicely, no matter how many atoms I'm depositing. And so, in a sense, it's a new kind of chemistry in that there's not one answer, but when you're down in this region, it's a stochastic chemistry. It's a chemistry that has variable numbers of atoms in that. Once you get to depositing 9, 10, 11, now you start to get a much more uniform probability with one peak that's close to the true one. So this was the So what I had just shown is a story of platinum for reducing protons, and that is a kind of single atom, single metal reaction. And now the question could be, which I'll now try to answer, what if I just don't do a metal, but I do something much more interesting, which is make a molecule, and I do 1 molecule, 2 molecules, 3 molecules. And these molecules are oxides of a metal. So instead of putting down a single metal atom, I put down a metal oxide. And so what's being shown here is a catalyst that's been looked at a lot in recent years for oxidizing water. And these are cobalt oxide catalysts. They're quite good catalysts compared to much more expensive materials. And so you can see, for example, the one on the upper left corner is just one cobalt atom surrounded by a number of oxygens, some from water and some just as oxide. And that's a single atom molecular catalyst, because a single atom forms a molecule. If I put two atoms, then you get the one over on the middle, and so on. And so I have the same kind of experiment, but here I'm growing one kind of a molecule, which is one atom of cobalt, two atoms of cobalt, three atoms, and on top of this, I have oxide surrounding the whole catalyst. And so now the question is, does this follow the same rules? If I now put down one of these, or two of these, or three of these, is it the same that one is the least active catalyst, and two is better, and three is much better, and so on, until I get up to a bulk cobalt oxide that I synthesize and put on an electrode? And so we do the same kind of an experiment. It shows the two steps. I first deposit, I take cobalt ion in solution, say at pH 7, and I oxidize it from cobalt 2 to cobalt 3, and when I produce the cobalt 3, it forms an oxide, and it deposits on the electrode. And again, I'm working at very, very low concentrations, femtomole, 10 to the minus 15 molar concentrations. And the same strategy of using such a low concentration that I have a very, very few atoms in 10 seconds or something. And then I put it into a solution where this has a much more alkaline pH, and water will be oxidized. And now I get a much larger current than from that femtomolar, and that shows me the oxidation of water on these molecular catalysts. And so it's basically the same kind of reaction that I talked about for platinum and proton reduction, but here I'm growing these molecules and seeing if the molecule behaves the same. So the next slide shows the same thing now for a series of cobalt oxides. And again, you can see that as I increase the number of cobalts, the current is going up. Now the oxidation current we're plotting is going down. So the current gets bigger and bigger. But the location of that wave is not changed very much. And so if you look at this one on the right, we'll show it on the next slide, I think is even better, you can see a whole series with rates, and you can see how the rate is affected by the size. And it doesn't change very much. In other words, the basic picture here is that one molecule is just about as good as 2 or as 3 or as 4. In other words, here you don't see this very bad single, and then it gets better and better as you make an ensemble. In this one, you see it improving. You can see it improving, but very sharp. That one is almost the same as the others. And so a molecule by itself behaves now like a bulk catalyst. And it's a different picture, a molecular catalyst compared to single atom metal catalysts. And so you can see this same trend. The next one shows the same stochastic that's seen. If you do few atoms, you get this distribution of sizes, and that fits very well, which gives us confidence that what we think we're seeing is right, because this is clearly a difficult experiment, and one that has generated some controversy. So I think the message I would like to leave you with is that I think we're in kind of a new era of chemistry, which is chemistry of single entities. Entities can be atoms, they can be nanoparticles, they can be molecules, but that we do experiments not by having billions and billions and billions of these and averaging out all the behavior, but single ones and studying the properties of single ones and seeing if we can learn from that how to build catalysts and what the real basis of why these materials are catalysts, why they're catalysts for hydrogen reduction, water oxidation, and so on. And that's, of course, fairly fundamental in terms of molecular catalysts to a lot of biology where we depend totally on proteins that are enzymes and that catalyze many, many reactions, and trying to understand how that little group in them does the reactions, because the biologically evolved catalysts are always much, much better than the ones that we can try to design. For example, if you think of a catalyst for evolving oxygen, which is an important reaction, if we make a manganese oxide catalyst, which is the basis of the water oxidation step with photosystem 2, it's never as good as the photosystem 2 itself, which is a very, very effective catalyst. So nature and evolution over many, many years has produced some of the best catalysts by just trial and error. If we can understand a little better, maybe we can now produce them and design them to be even more active. So if I can go back to the first slide. Back to the first slide. Okay. So this is my story. I want to give credit to the people who really did all the hard work. And so the names of the postdocs there, Min Zhu and Xiaoyo Jin. Min did the work on the catalysis, and Jin did the work on the molecules. And they're both very, very talented students who did very difficult experiments. And I think are perhaps showing that there is another way of doing chemistry than the way we've done it for so many years. Thank you very much again for the invitation. I hope that this was intelligible from the point of view of PowerPoint slides. I had hoped I could draw pictures on the board and tell the story the way I teach my class, but not in a classroom. So thank you very much.
Thank you. Thank you.
Okay. No questions? Okay. Okay. No, that's fine.
Thank you very much indeed, Professor. It's really very valuable lecture and very valuable information. And because we are running of the times, we'll keep the question at the end of our lectures. Now we will try to introduce Professor Olsen. Was born in Norway in April 1940. He received his medical and doctoral degree from the University of Oslo in 1967, where he becomes a faculty member of the Electrochemical Institute and conducted molecular study on the structure of collagen in 1971. He moved to the United States to work with the Professor Darwin, and 1 year later joined the faculty of the Department of Biochemistry at Richard Medical Schools. In 1985, Professor Olsen was appointed the Hersey Professor of Anatomy and Cellular Biology at Harvard Medical School. Now he is at cell biology in the same department. Since 1996, he has also been senior member of the staff of Washington Institute and professor of the Department of Biology at Harvard School of Dental Medicine, where he was Dean for the research from 2005 and 2017. The floor for you, Professor Olsen.
I feel honored for staying here, and I hope you will like what I will tell you about the skeleton and how it works. Now, obviously, the skeleton is absolutely essential for us to sit here, and it's remarkable how the cells that form the skeleton are able to put this wonderful organ system together during development and change it as we grow, and then finally keep it going until we are very old. And as a medical student, I was many years ago really fascinated by the molecular biology, the cell biology, the genetics that really makes this possible. And I decided that as a scientist, I would take advantage of the mutations that occur in different proteins and different genes that affect the skeletal development and function to really understand the mechanisms, the molecular, the cellular, and the biochemical mechanisms that makes this possible. And many years ago, in 1996, I had a postdoc in the lab, Matt Warman, who learned about a disorder called osteoporosis pseudoglioma syndrome. And these are individuals that have too little bone, almost no bone in certain places, and this is combined with blindness. And by getting patients with this syndrome from many different collaborators, we were able to map the position of the mutant gene in a specific chromosome, chromosome 11 in humans. And once we had mapped the position of this gene that made formation of the skeleton impossible at certain places, and you can see here below, on the left side, at the very bottom, you see the bone, the pelvic bone in a patient with this disorder is very thin, like paper, compared with above it, the pelvic bone in a normal individual. And we were eventually successful in finding the gene that causes this syndrome of loss of bone or very little bone. And this gene coded for a receptor that sits on the surface of bone-forming cells. It's called LRP5, and you can see that. And this is a receptor that binds secreted glycoproteins called Wnt proteins and a frizzled protein that sits on the cell membrane. And when LRP5 forms a complex with this Wnt protein and the frizzled protein on the cell surface of bone-forming cells, then the cells become osteoblasts, or bone-forming cells. So this decided that we wanted to understand what the downstream signaling was and what this would do. And so this is a case of osteoporosis, genetic, that is caused by this LRP5. Now, it turned out that LRP5 also could be integrated and form a complex with another protein called sclerostin. And this sclerostin, when it binds to LRP5, prevents LRP5 from forming the complex on the right and signaling an osteoblast formation. So in the cells that produce bone, there are LRP5 signaling pathways that form bone, or as you can see at the bottom. But in addition, cells producing sclerostin put a brake on the activity of LRP5 and bone-forming cells. And this sclerostin makes certain that we do not have too much bone, but just the right amount of bone. And this was discovered at about the same time that with mutations in sclerostin, so that sclerostin cannot bind to LRP5, then you end up with individuals with too much bone. So much bone, in fact, as you can see on the left, on the bottom, that one skull of a patient with mutations in sclerostin leads to sclerosis, and a bone in your skull that is as heavy or heavier than 3 normal skulls. So here we have, with mutations in LRP5, you get too little bone. With mutations in sclerostin, you get too much bone. And this has led very recently to a new drug to prevent or use in osteoporosis. Because it was clear from this that if one had an antibody that would bind to sclerostin, normal sclerostin, and prevent its binding to LRP5, one would have a drug that would increase bone formation. And you may know, if you're interested in bone biology and osteoporosis, that this drug, an antibody to sclerostin, is now approved for use in patients in the USA and in Japan. And it replaces many other drugs that have been used for a long time for treatment of osteoporosis. Now, at the time when we discovered some of the pathways that lead to bone formation, we also discovered in patients that bone can go haywire. Bone formation can, together with inflammation, really destroy bones. And this mutation I show here leads to a disorder called cherubism. It's a genetic disorder with excessive inflammatory bone loss in children in the jaws, in the mandible and the maxilla, and in not other bones. And it was clear to me that if we could find the mutation that resulted in this disorder, we would learn something specific about what controls the formation of the jaws that we all have, the mandible and the maxilla. And in this disorder, you can have familial and sporadic cases. Most of the disorder, the cherubism, are produced by dominant mutations, but there are rare recessive cases. And the onset of this disorder starts pretty soon after birth, around 1 to 2 years of age. So in the period between 2 and 4 years of age, this disorder takes place. And remarkably, after puberty, some of the effects of these mutations regress, leaving some facial deformity or radiological evidence. So again, having these patients, finding the gene would tell us something about what controls the bone formation in these specific bones in the jaws, as well as what would happen during aging when these children with these mutations would grow up. And if one looks at pieces of tissue from patients with cherubism, you also see, as shown here on the right top, there's inflammation in the tissues that normally should have the bone. And within these inflammatory fibrous tissue regions, you see a little bit of bone, and you see multinucleated giant cells. And these giant cells are remarkable. You see them here in the hard modification. These are cells that have 5, 10, 20, sometimes 50 nuclei in a single cell. These are not the normal cells we have in our bones. And as a consequence of these cells, the bones in our jaws are eaten up by these multinucleated giant cells, the osteoclasts, bone-eating cells. Now, it was known at the time when we looked for the mutations responsible for this disorder that myeloid cells in the bone marrow, shown here from the very bottom and up, form the cells that over time develop into macrophages and osteoclasts. And for the formation of macrophages and osteoclasts from myeloid cells in our bone marrow, there are certain cytokines and receptors that must be activated. And so a macrophage colony-stimulating factor, MCSF, is needed to form the macrophages. And together with that cytokine, you need another molecule called the rank ligand to bind to a rank receptor on the cells to form osteoclasts. And we were wondering whether the mutations in cherubism could actually explain what the connections might be between these different steps in the formation of osteoclasts. And also tell us something about how bone marrow stromal cells regulate the formation of macrophages and osteoclasts. And it turns out that in addition to having factors that can produce osteoclasts and macrophages, stromal cells in the bone marrow produce proteins. Osteoprotegerin is one of them, that prevents that process. And again, this is a system where you have cytokines that stimulate osteoclast formation, bone resorbing cells, but also cytokines and molecules that inhibit, again, to make sure that we do not get too much osteoclasts or too little, just right, as if you have in the system that produces or stimulates the production of bone by bone-forming cells. So we were able to map the gene for cherubism on chromosome 4, at the very tip of chromosome 4. And it turned out that that part of chromosome 4, when we discovered this, had already been sequenced. And the sequence was quite good. But in the region of that gene, we had 25 genes. And so to find the gene that was mutated in cherubism, we started sequencing one gene after another. And that took some time, but we identified the gene in 2001. And we had 15 families with cherubism, mostly from Brazil, but we found mutations in 12 of these families. Now, these mutations were in the protein that had the not so interesting name, SH3 binding protein 2. It's a protein that is produced by a gene with 13 exons and separated by introns. And all the mutations that we had in these patients were mutations that changed an amino acid in exon 9. And the mutations in all these families we looked at changed an R, P, G to something else. R for arginine, or proline, or glycine, very close together within the sequence of 6 amino acid residues in this protein. And they were replaced by either glutamine or proline, leucine, arginine, histidine, etc. And we did not find any mutations elsewhere in this protein. And it turned out that when we looked at the protein, it had many different domains that could interact with other intracellular proteins. But these domains were not carrying mutations. All the mutations were in one region, outside any region that looked like the regions interacted with other proteins. And so the question then was, how could these mutations affect the functions of this protein? Well, the more we looked at the protein, we more found that it really is interacting with many different proteins in practically all cells, but especially in bone-forming and bone osteoclast-forming cells. And its position or its activity activates multiple signaling pathways in the protein, in the cells that express this SH3BP2. And we then decided that in order to understand how it works and what the mechanisms are really for cherubism, we had to make a mouse model. You cannot do experiments with humans, so we tried to do it in the mouse. Put one of these mutations that we found in the patients into mice. Now, in doing that, many people working with mice at the time would almost immediately knock the gene out. And I'm very glad that we did not do that. Because what we did find was that there's another syndrome, Wolff-Hirschhorn syndrome, that have deletions in chromosome 4. And frequently, the deletions in this syndrome include the gene SH3BP2. And none of these patients had cherubism. So clearly, we should not knock out the gene for SH3BP2 to get a mouse model for cherubism. We had to take one of the mutations in cherubism from humans and put that type of mutation into the gene of the mice. And we did that. And that gave us a mouse with cherubism. Except that when the mice carried only one allele with a mutation, they were losing bone, but they did not have inflammation. However, when they were carrying 2 alleles of the SH3BP2 with that specific mutation, their whole body turned into an inflammatory bone-losing structure. as if the whole body of the mouse was a copy of what happens in the jaws of people with only one allele mutated in this protein. And so, what happens in the jaws of people with cherubism happens in the whole mouse with carrying the mutation in the 2 chromosomes of SF3B2. If we have one of the 2 alleles mutant in mice, we get aggressive giant osteoclasts with multiple nuclei, but we do not get inflammation. So we have looked at these mice. As one would expect, the mice carrying the mutations in both alleles on the top right lose bone. And the bone loss is caused by osteoclasts that are abnormally large. They have, instead of 4 or 5 nuclei per cell, they have up to 50 or even more nuclei in cells. And these cells sit on bones, and they really eat the bones up in the mice. And as you can see here at the bottom, in the mice, there are increased numbers and increased sizes of osteoclasts going from the wild-type control on the left to the double mutant mice on the right. Now these osteoclasts are not only containing many nuclei, but they are giant cells. And that can be shown in looking at mutant bones. This is the maxilla of the mouse, cherubism animal. And on the right, you see in dark color here these osteoclasts that are sitting on the surface, and they are absolutely abnormal. And not only are they abnormal in the mouse, but when we take bone marrow cells from normal or mutant mice and add the proteins that are needed for their differentiation to osteoclasts, then we see that in mice or in cells from mice with cherubism, we have these gigantic osteoclasts. And this you can see better here. So on the top, these are normal myeloid cells stimulated to become osteoclast. And you see the osteoclast on the top here, they're very small cells compared to the mutant cells. Mutant cells are gigantic, and the number of nuclei really is proportional to their activity as bone-eating cells. And not only are these cells eating bone, but the inflammation and macrophages in inflammation start eating bone from the outside, not only from the inside. So in cross-sections of a femur of the control mice and mutant mice on the right, when you have 2 copies of the mutant SH3BP2 in mice, the bones are eaten from the inside by osteoclast, but also from the outside by inflammatory macrophage cells. And this is in the mouse. It's not only macrophages that invade and eat bone, but there are macrophages that go through all organs, including liver, lung, and stomach. And what you see here on the left are in these different tissues staining for macrophages that have invaded the liver and the lung and the stomach in the mutant mice. And what we discovered was that not only are these tissues invaded by activated macrophages, but these macrophages produce tumor necrosis factor alpha, TNF-alpha, in large amounts. Normally in mice, you cannot detect this in the serum, but in the mutant mice, shown here on the right, you have very high levels. So these macrophages that invade the organs and eat the bones spell out really large amounts of TNF-alpha, and as a sign of highly activated macrophages. And these highly activated macrophages invade joints that destroy joints, shown here in the elbow joint, comparing the wild type and the mutant joint, and the knee joints, as if you would have absolutely no respect for the bones here, but cells that try to, as much as they can, destroy the bones and the joints between the bones. So this led us to put together a hypothesis for what mutations in SH3BP2 can do. It happens in myeloid progenitors, and because of the mutation, these progenitors become overly sensitive to incoming signals. And you get activated, highly effective, and TNF-alpha producing macrophages on one part, and that leads to inflammation and bone loss. In addition, you get hyperactive osteoclasts coming from the same myeloid cells that really, on top of that, destroy the bone. So it's said that SH3BP2 is important for the balance and the response of incoming signals in myeloid cells and the formation of macrophages and osteoclasts. Now, we looked at in the mice, we could, of course, determine some of the signaling pathways that were affected by these mutations in osteoclasts and macrophages. And in doing that, we found that TNF-alpha production is up because of specific signaling pathways, ERK1/2 pathway, for example, that stimulates TNF-alpha production. And since we had this high TNF-alpha production, we obviously thought that if we knock TNF-alpha out, we remove that gene from the mice, perhaps they would be normal. And we did that. We took the TNF-alpha gene and produced mice that were deficient in this gene, and TNF-alpha, and we improved the bones of the mice. However, what we did find was that the invasion of macrophages into liver, lung, and stomach, shown here, without TNF-alpha, disappears. The inflammation disappears. However, when we look at osteoclasts, there's no difference whether you have TNF-alpha gene active or not. So in the diagram that I showed you, that SH3BP2 in myeloid cells leads to macrophage activation and osteoclast activation. If one takes TNF-alpha out of the mouse, then the macrophage arm is normal, but the osteoclast arm is not. So there we looked at what determines the high activity of osteoclasts. We found that downstream of the mutant SF3BP2, there's a protein NFAT-C1, which controls osteoclast differentiation. And this was upregulated in the mutant mice. And NFAT is an inhibitor of the osteoprotegerin that in turn reduces osteoclast differentiation. So with NFAT-C1 being up, then osteoclast differentiation would go up. So then we decided that we would simply knock NFAT-C1 out and see whether that would take care of the osteoclasts. And in fact, that happened. In these mice without NFAT-C1, we had no longer osteoclasts, seen on the right here, that are abnormally large and very active. We have normal osteoclasts. However, the invasion of inflammatory cells, macrophages, into the lungs or other organs is now not normal. So we have two arms, and we can play with proteins within the two arms to reduce the bone loss in cherubism. But then, with all this, we still do not really know why mutations in a very small region of this protein can affect all its interactions with other proteins and signaling pathways. How could that be? And these mutations are in a very tiny portion of SF3BP2. And it shouldn't affect any other part of the protein. And all the other parts of the proteins that are seen in green and red here in the diagram, these are the parts that really stimulate signaling pathways in cells. So what is these mutations on top here? What are they doing? And this was solved with 2 papers in Cell, showing that this protein, like many proteins, they can bind to tankerase. Tankerase is a poly-ADP ribose polymerase, and it binds to specific proteins in cells. And when it does that, it puts a signal on other parts of the protein. And in this part, it's one of the parts that are involved in signaling pathways, interactions with SF3BP2. And this signal put in by binding of tankerase to SF3BP2, that leads to ubiquitination and destruction of the protein. So what is abnormal in these mutations in cherubism is that it is not degraded. It builds up in cells, so the level of expression of this specific protein is too high. So that what really causes cherubism with these mutations in SF3BP2 is that there's too much of it. There's nothing really wrong with it, but it's simply too much. And so the region in SF3BP2 that allows it to bind to tankerase, the question is that region shared with other proteins? And in fact, one of the 2 papers shown here shows that there might be 200 or more proteins that carry a small region similar to the region that has mutated in cherubism in SF3BP2. And that in these proteins, any mutations in this kind of small region might again affect its binding to tankerase and therefore destruction, which is the normal way of making sure that the levels of the proteins are not too high and perhaps not too low. And so, having done this, we still were wondering, why in humans are these mutations primarily affecting the jaws, the mandible, and the maxilla? And so, looking at what happens from birth with these two bones, both the mandible and the maxilla at birth are simply rod-like shaped proteins. Within 4 to 5 years, these proteins are remodeled enormously. And to remodel the protein and reshape the protein, you have to degrade the protein, the osteoclast, and activate osteoblasts and form bone. So, at 4 to 5 years of age in humans, these two proteins are the only proteins in the skeleton that are almost fully sized and shaped. And so this activation of these specific proteins might be one of the reasons. But there might be something else. And that is that cherubism, when it occurs in patients with mutations, happens mostly before 5 years of age. Then it starts. And what is unique, in addition to the remodeling early in life of the jaw bones compared with other bones, is that these two bones have macrophages that are responding to oral bacteria and tissue fragments caused by jaw remodeling and tooth eruption. And in a paper with my postdocs and other collaborators as well, we published in 2014 this cell report paper showing that what initiates the cherubism happens in the jaw bones because the macrophages are activating with the mutation SF3BP2. They activate what is called the toll-like receptor, the immune system that we all are born with. And what happens here is that in addition to having macrophages on the left here, TNF-alpha producing macrophages with a mutation in cherubism, these macrophages in the jaw bones increase the amount of what is called PAMPs or DAMPs. These are oral microorganisms and jaw tissue fragments that can bind to the toll-like receptors shown here in the red. And by binding to these receptors, you activate another signaling pathway in macrophages that produce more TNF-alpha and add to the process. And within this part that really develops the cherubism phenotype, there is downstream of the toll-like receptors a protein called MyD88. And if this is correct, one should expect that by knocking out MyD88, that one would reduce the effects of cherubism. And this is exactly what happened. So now we made a mouse that had not the cherubism mutation only, but in addition lacked the MyD88 shown here. And in these mice, the TNF-alpha production goes down to zero, normalized, the effects of liver, lung, or macrophages getting in and causing inflammation and inflammatory loss of tissue, that's normalized. So clearly, for cherubism and the mutations to really work, certainly in mice, and we think in humans, you need, in addition to the original mutation in the macrophage, you need the toll-like receptor activities in the jaw of the patient. So that leads me to the last question I want to address, and that is, why do the disorders of cherubism regress after puberty in humans? Now, it turns out that this toll-like receptor effect in macrophages in the jaws, not only do they have MyD88, but another protein, IRAK4, with mutations in MyD88 or IRAK4, you get in humans another disorder called primary immunodeficiency. And this is particularly susceptible to pyogenic bacteria. Now, in people, not with cherubism, but with this primary immunodeficiency, after puberty, as they grow older, it doesn't seem to matter anymore. They don't die. If they die of the immunodeficiency, it happens before they are 11 or 14 age. And so afterwards, after puberty, this system involving toll-like receptors and downstream signaling pathways seem to be turned off. And I think that is a good chance that this is why in patients with cherubism, you get regression after puberty. This, of course, has to be studied, and we're not can do that in mice, but you have to do it somehow with patients. And finally, do these mechanisms point to effective therapies for patients with cherubism? Yes, I think we do. So in looking at the critical parts in the signaling pathways that cause the cherubism disorder, you can affect the TNF-alpha production, number one. You can affect rank ligand and the formation of osteoclasts, number two. Or you can affect NFAT-C1 that has to do with differentiation of osteoclasts. And all three things have been tested in humans. And in fact, it turns out that antagonists of TNF-alpha, rank ligand, and NFAT-C have been tested. And the outcomes is that for, number one, TNF-alpha is not sufficient. It doesn't really fully work in patients. However, if in mice you take away TNF-alpha at the very beginning of the mice, before they get cherubism, that prevents it. But that's hard to do in humans. The rank ligand effect have been tried. It doesn't really work either. It doesn't really cure cherubism. However, affecting the NFAT with a calcineurin inhibitor, calcineurin is absolutely central for the effect of NFAT, that has been very good. And in fact, that you can see here, before treatment on the top left, next to after treatment of a 4-year-old boy, for 1 year on the therapy, it's almost rectified. So I think we do have now a way of treating patients with herbism. Now, all these studies, starting from the very beginning, I would say is due to 3 postdocs, Yasuyoshi Ueki from Japan, Valdenise Tiziani from Brazil, and Ernst Weichenberger from Germany. Now, Ueki, he got his PhD degree in the laboratory of Professor Yoshifumi Ninomiya at Okayama University. And Ninomiya was my first postdoc when I came from Norway to the USA. And he returned then to Japan. So he's my or he's my son, Ueki. And in addition to these 3, everything I've talked about in the different aspects of this specific disorder included all the different names that you see here. And it would not have been possible without these collaborations between different countries and within a country. And it wouldn't be possible without funding from the National Institutes of Arthritis and Musculoskeletal and Skin Diseases at NIH in the USA. And thank you for listening. Thank you.
Thank you very much, Professor. This is very valuable lectures and also information. I would like to apologize for the audience for the tight schedule of the scientist. I will not be able to take any questions, but we will have time in the lunch at 12:30 in College of Science. If you have any discussion with the professors, you're most welcome. And thank you very much indeed. Thank you for all of you. Thank you for the scientists. Thank you very much indeed. Thank you.